Sydney. She completed a Bachelor of Advanced Science with honors in 2020 and studied new techniques to probe model protocell permeability for her undergraduate senior thesis. She began her PhD in 2021 and has continued to study model protocells, focusing on how they can access the nutrients required for continual membrane growth, with the goal of building a synthetic cell that can grow and divide using the simplest components possible. So without further ado, you can take it away, Lauren.
Thank you very much. I will just share my screen. Is that looking okay?
Looks good.
Great, thank you. Well, hello everyone, and thank you for the introduction, Becca. My name's Lauren, and as you said, I'm a final year PhD candidate in Anna Wayne's group at UNSW Sydney in Australia. And today I'm going to tell you all about some of the work that I've been doing on model protocells or model primitive cells. So to start off with, I just thought I'd begin with the acknowledgements. First, I'd like to acknowledge the entire Wayne group and especially my supervisor, Anna, for all her support throughout my PhD. I'd also like to acknowledge the Australian government and the Human Frontier Science Program for funding my research. So to begin, I'm sure many of us here are trying to at least somewhat answer the question of how did life on Earth emerge? Now, I'm trying to focus on understanding how maybe the first cells emerged on Earth. As we all know, modern biology itself is extremely complex. Even really simple cells, have a lot of complexity going on with them. And this complexity would not have been possible when life was first forming on Earth. So perhaps a simpler version of modern biology would be a phospholipid-based protocell or primitive cell that maybe has a simple phospholipid bilayer membrane, like what our cells are composed of, and maybe has a few simple proteins and enzymes, and perhaps there's some genetic material inside. But even these phospholipid-based protocells are actually quite complex. As I said, proteins themselves can be quite complex in nature and probably wouldn't have existed on an early Earth environment. And phospholipids usually require dozens of enzymes in order for them to actually be synthesized. So that was pretty unlikely to have happened in an early Earth environment as well. So we need an even simpler version of a protocell to perhaps accurately model what the first primitive cells on Earth may have looked like. And one form of this may be a fatty acid-based protocell. So rather than the membrane being composed of double-tailed lipids like phospholipids, we have a single-tailed fatty acid. And these are much chemically simpler than phospholipids. They can be formed abiotically and have been found on meteorites. So it makes them potentially plausible candidates for forming the first membrane-based protocells on early Earth. So today I'm going to talk you through some of the work I've done on fatty acid-based protocells, and particularly how their properties are affected by really small shifts in pH. So just to begin, as a quick recap for everyone, bilayer membranes are usually composed of lipids, and in this case here, we're talking about fatty acid-based bilayer membranes. And the reason these bilayer membranes form are because lipids are composed of a hydrophobic region and a hydrophilic region. And that allows them to self-assemble into aggregate structures such as spherical bilayers or vesicles, similar to what our cell membranes are like. Now, fatty acids are a little bit unique in the sense that a lot of their behavior is quite pH dependent. And this isn't really the case for regular phospholipids. So a simple fatty acid like oleic acid, which is just an 18 carbon chain fatty acid, looks something like this. However, when you increase the pH, it actually becomes deprotonated and the head group becomes negatively charged and you form oleate. Now, this pH-dependent behavior actually enables vesicle formation to occur for fatty acids. So when you have approximately half of the molecules being protonated and half of them deprotonated, it leads to the formation of bilayer membranes or vesicles. And this occurs at what's known as the apparent pKa of the fatty acid, which is roughly 8.5 for oleic acid systems. Now, if we were to shift significantly below the pK of the fatty acid, all the molecules would become protonated, and that would just result in an oil forming. Conversely, if we were to shift super highly above the pK of the fatty acid, we would actually lead to all the molecules become deprotonated, and that would lead to repulsion between the head groups and the formation of micelle-like structures, which are basically just soap. Now, this pH-dependent behavior is pretty well established, but what's less well understood is just looking at the pH range within which vesicles form, which is roughly 1 pH unit outside of the pKa of the fatty acid. A really small shifts in pH, actually within this pH range, do they affect the properties of the vesicles? This hasn't really been well studied prior to my work on it, even though this entire system is largely dependent on pH. So yeah, I wanted to explore how fatty acid vesicles were affected by small shifts in pH, and consequently how fatty acid-based protocells may be affected by small shifts in pH. And the properties I was interested in investigating were first looking at how the lipid molecules packing together is affected by pH, and then looking at how this lipid packing could lead to changes in permeability or changes in rupture tension. So to begin with, we'll focus on lipid packing. And in order to better understand how lipid packing changes with pH, I prepared a bunch of oleic acid vesicles at varying pHs and embedded a fluorescent probe known as Louden into the membranes. So Louden is a probe that's used to actually measure membrane polarity. Its emission spectrum is dependent on basically how much water is around it. So we can see with increasing water around the probe, there's a change in the emission spectrum. And we can just monitor this using some sort of fluorimeter. So as I said, what I went on to do was prepare oleic acid vesicles at a variety of pHs and explored how this emission spectra changed with that changing pH. As we can see in the plot on the bottom here, there's kind of two peaks that are prominent for fatty acid vesicles. We have one here around 430 nanometers. We have another one around here at 500 nanometers. And it's quite clear that as the pH increases, there's definitely a change in those spectra there, with this peak here at 500 becoming more prominent for the higher pHs. So that's telling us that something is going on with membrane polarity there. And we can actually get a more quantitative measure out of this data by pretty much comparing the peaks at 500 and the peaks at 430 to determine what is known as the generalized polarization, or GP, which is pretty much just a ratio of those peaks. And a higher GP means that the probe is more solvated. So we're looking at bilayer structures that have increased water penetrance actually within the bilayer. And we can see in this plot here that as pH increases, there's definitely an increase in GP. As I said, what this means is that there's increased water penetration in the bilayer. Now, there could be a couple of reasons behind this. One could be due to there being increased repulsion between the lipid head groups, because as the pH increases, they're more negatively charged. And also as pH increases, we see the formation of more higher curvature structures, which could allow water to actually pass into the membrane a lot more easily. But in order to kind of better visualize what might actually be going on here, we turn to doing some molecular dynamics simulations, which were performed by our collaborator, James. And what he did was simulated octanoic acid bilayers at 50% protonation and at 25% protonation. And here we can see a top view, so looking down on the bilayer, and a side view of the bilayer as well. And what we were able to find is that as we decrease protonation or we increase the pH, there's obviously a significant difference in the way the bilayer looks. It becomes a lot more disordered at higher pHs. There's a lot more gaps in the membrane. There's a lot more defects. And we also see an increase in the area per lipid. So each lipid molecule is starting to take up more space. So we're seeing some pretty significant changes in membrane packing here. And this could possibly explain why we saw increased water penetrance with higher pHs. It's likely owing to this increased membrane disorder. So there's more gaps, more defects in the membrane. It allows water to pass through more easily and actually enter the bilayer itself. So continuing on, we wanted to probe one more aspect of the lipid packing, and to do that, we used a technique called electrical impedance spectroscopy, or also known as EIS. And basically what this technique involves is tethering a lipid bilayer to a gold electrode, and then applying alternating current across the membrane. And what this pretty much allows you to do is to measure some of the electrical properties of the bilayer. So something like conductance, which is proportional to the ion flux through the membrane, and then capacitance, which is inversely proportional to membrane thickness. So to start off with, I'm just going to focus on the capacitance, which can be used as an indicator of membrane packing because of this change in membrane thickness. So again, I worked with an oleic acid system, and I prepared oleic acid tethered bilayers this time at a variety of pHs and subjected those bilayers to changes in pH and explored how the capacitance changed with those pH changes. And in this plot here, we can see that with increasing pH, even with the pH shift of 0.5, which is actually quite small, we do see that the capacitance increases. So that's telling us that as the pH decreases, the thickness of the bilayer, as the pH increases, the bilayer thickness decreases. So we're seeing the bilayer become thinner at higher pHs. So overall, if we combine this with all the other results that we've seen in regard to lipid packing, we're starting to build up a picture of how just small shifts in pH can change the way the lipid molecules and fatty acid bilayers pack. With a slight increase in pH, we've seen that we can get a thinner bilayer, we can get increased membrane disorder, and we can get increased area for lipid. And it is expected that all of these changes in packing would likely affect the actual biophysical properties of these vesicles, of these fatty acid-based protocells. So I wanted to explore a couple of these properties that would likely be affected by lipid packing. And the first one I'll start with is permeability. So in order to look at how permeability perhaps changes with pH, I again turn to the electrical impedance spectroscopy measurements. This time, rather than looking at capacitance, I focused on conductance, which, as I said earlier, is proportional to the ion flux through the membrane. So it's giving us an indication of how easily ions are flowing through the membrane. So it's telling us a bit about ion permeability. And again, using the same system as before, I worked with oleic acid bilayers, and I exposed them to changes in pH and explored how the conductance changed with those changes in pH. And I was able to find a pretty clear trend that showed that with increasing pH, we do see an increase in conductance. So that's telling us that there's increased permeability to ions with increasing pH. Now, this is probably not that surprising when we consider the information we've learned about the lipid packing of fatty acid bilayers. We've seen that with increases in pH, first of all, the membrane gets thinner, and various studies have proposed that permeability can be inversely proportional to bilayer thickness. So solutes can often permeate the bilayer more easily if the bilayer is thinner. So perhaps not that surprising that we've seen an improvement in ion permeability with increasing pH. But what's probably more likely is that we've seen an increase in ion permeability due to the increased membrane disorder. So similar to how I spoke about how water may have penetrated the bilayer more easily because there's more defects, I think a similar thing is probably happening here. Because of the increased number of defects, or essentially gaps and holes in the membrane, it allows small ions such as sodium to pass through much more easily than they would when there's no defects present in the membrane. But continuing on with this, I wanted to explore whether or not this permeability trend could be seen for other solutes. perhaps solutes that are much bigger than just a sodium ion, for example. And in order to do that, I looked at the permeability of a fluorescent dye known as HPTS, which is basically a dye that gets incorporated into the aqueous interior of these vesicles, as demonstrated by the yellow here. Now, this dye it's quite large. It's a lot bigger than a sodium ion, that's for sure. So we would expect that it would permeate, if it does permeate at all, it would permeate a lot more differently and probably have a much more difficult time actually crossing the lipid bilayer. So in order to see whether or not perhaps increasing the pH would make the permeability or the permeation of that HPTS dye molecule easier, I looked at oleic acid vesicles again in a variety of pHs, encapsulating this dye, and I looked at them under a epifluorescence microscope. They look something like this image here. So you can see them quite clearly. And in order to monitor whether or not the dye was actually leaking out of the vesicles or if there was dye permeation occurring, I monitored the fluorescence intensity of the vesicles relative to the fluorescence intensity of the background. So we would expect that if the dye was leaking out, the relative fluorescence intensity would decrease over time. So as I said, I prepared oleic acid vesicles at a variety of pHs and explored both whether or not there was a difference in the relative fluorescence intensity for the different pHs. And I also looked at whether or not this fluorescence intensity changed over a period of about 4 days. And basically, what these plots show is that there didn't really seem to be a significant difference in the fluorescence intensity for vesicles prepared between a pH of 8.2 and a pH of 8.9. And this fluorescence intensity didn't really seem to change over the period of 4 days either. And that's demonstrated by the lack of change in the intensity over here, and also the lack of change in the gradient when we compare the gradient of these essentially slopes as we look at these plots with the varying changes in pH. So what that seems to suggest is that for larger molecules such as HPTS, that fatty acid vesicle permeability doesn't really change despite the small increases or decreases in pH. And that's not necessarily a bad thing when we're thinking about these vesicles in the context of protocells. So it would be important that protocells would at least be able to retain some of their material. So important molecules, say genetic material, for example, or other macromolecules would be necessary for the protocells to actually function. So we would need them to actually be retained within the vesicle rather than just leaking out all the time. But perhaps smaller solute molecules that could be useful nutrients or useful waste molecules, perhaps it's important for them to be able to permeate the bilayer much more easily. So again, overall, it's not necessarily a bad thing that these larger molecules don't seem to be able to permeate the bilayer, but smaller molecules, such as small ions, do seem to be able to permeate the bilayer, and that permeability is increased with increasing pH. So I started to further build up this picture of how subtle shifts in pH affect fatty acid bilayers or fatty acid-based protocells. And I now wanted to explore another property that is known to depend on lipid packing, and in particular, the presence of pore formation, in particular, increased pore formation, and also bilayer thickness is rupture tension. So rupture tension is pretty much a measure of how prone a vesicle is to rupture. How likely is it that it will burst? So in order to actually look at the rupture tension of fatty acid-based vesicles, I first prepared a bunch of fatty acid multilamellar vesicles, or also known as MLVs, again at a range of pHs, and then I subjected them to some pressure. And that, in this case, was in the form of extruding these vesicles. So extrusion is basically a process where you force these quite large multilamellar vesicles through a membrane with pores of a set diameter, and they're usually quite small. So in my case, I was using 100 nanometer pores. So they're going from some of them roughly 20 microns in size to being forced through little holes that are approximately 100 nanometers in size. So this pressure as these vesicles are being forced through those pores actually causes them to rupture, as demonstrated here, and then it leads to the formation of small unilamellar vesicles, or SUVs. And these are roughly all equal in size, and they're all usually unilamellar, or just have a single bilayer. So after subjecting these fatty acid vesicles that are at a range of pHs to this pressure, I then measured the size of the vesicles post-extrusion and found that with increasing pH, the size of the vesicles actually decreased. And this was measured using dynamic light scattering, also known as DLS. Now, this decrease in size actually can be used to determine the rupture tension of these vesicles, or at least approximate what the rupture tension may be and how it changes as a function of pH. So this equation here from a paper published by Paddy and Friskin shows that vesicle size is proportional to the square root of the rupture tension here, which is denoted by gamma. And here we just have the radius of the pores in the extrusion membrane, and the pressure P, the pressure these vesicles are exerted to. So if we use this equation to then determine what the rupture tension, the relative rupture tensions of these vesicle systems are, we get this plot here, which shows that with increasing pH, we approximately see a halving of the rupture tension at higher pHs compared to the lower pHs, which is actually quite a significant margin if we put this in the context of phospholipid-based vesicles. So another study found that for phospholipid-based vesicles composed of DOPC, which is a phospholipid with a double bond in each of the carbon chains, it roughly has a rupture tension of 10 millinewtons per meter. But if we were to add an additional double bond to each of these carbon chains, we see a halving of the rupture tension, which is roughly equivalent to the change in rupture tension we see just by increasing the pH by approximately 1 pH unit. So I think it's actually quite impressive how much the rupture tension of these vesicles can change with changes in pH. It's such a simple method, I guess, to potentially change the rupture tension. We don't actually have to change the chemistry of the vesicles themselves. We're not adding any double bonds or anything like that. It's just a super small shift in pH can lead to such a significant change in the rupture tension. So I was able to confirm this by preparing much larger giant unilamellar fatty acid vesicles, again at a range of pH. And then rather than extruding them, the form of pressure I exerted them to was through an osmotic shock. So osmotic shock, we can see for the higher pHs, cause the vesicles to become smaller and rupture, and they're much smaller compared to vesicles at the two lower pHs that I looked at. So what this is overall telling us is that vesicles at higher pHs are much more prone to rupture. They're much more likely to burst compared to vesicles at lower pHs, which may or may not be a problem when we consider this in the context of protocells again. If our protocell is subjected to a slight increase in pH and it does rupture and leads to the formation of much smaller vesicles or vesicles that disappear entirely, that's obviously not ideal. That would result in them losing a lot of their contents, would probably prevent them from functioning as a successful protocell. So in the context of origins of life research, perhaps these subtle increases in pH might not necessarily be a great thing for the ability of these protocells to retain their material. But in saying that, if we combine these results with the results we've already looked at, we've so far been able to determine that when the pH is significantly below the pKa or at low pHs, vesicles are able to kind of retain their material and they're a lot less permeable to small solutes. But when we increase that pH and we're well beyond the pKa of fatty acid vesicles, they have a much harder time retaining their material, and there is an increase in the permeability of smaller solutes there. So just to kind of wrap that story up, we've found that with minor increases in pH, we see increased water penetration and increased pore formation, and this likely relates to the increased permeability of ions at higher pHs, and it also relates to the fact that these vesicles are more prone to rupture at higher pHs. And again, this has pretty far-reaching consequences in terms of the ability of fatty acid vesicles to encapsulate things, both within the origins of life field and protocells research, but also just using these fatty acid vesicles for other systems, such as for drug delivery, for example, or for just studying soft matter systems in general. But specifically in regard to origins of life research, the increase in pH leading to an increase in permeability may be a good thing in regards to the nutrients and waste molecules permeating the vesicles, but may not be a great thing in regards for these vesicles to retain some of their material. So I'd just like to note that most of this work has been published in a paper in Soft Matter, talking about the effects of these subtle changes in pH. There's a bit more detail in there, so feel free to check it out if you're interested. And I also thought I'd just briefly mention some of the related work I'm doing that is, I guess, somewhat similar to the work I've spoken about today. And one of those projects is looking at the effect of cationic species on fatty acid monoprotocells. As you can see in some of these microscopy images here, different cationic species have quite different effects on fatty acid-based protocells. And I'm also looking at how things like membrane composition can affect phospholipid model protocell permeability. So jumping a bit ahead, I guess, in terms of like potential evolution timeline, how can changes in the membrane composition actually affect the properties of a phospholipid-based protocell? So if anyone is interested in either of those projects or any of the work I've spoken about today, or just wants to chat about protocells in general, feel free to reach out to me. My email is down the bottom there, and I'd also be happy to answer any questions later. Thank you, everyone, for your attention.
Thank you, Lauren. And I'm sure we're looking forward to asking you questions at the end when we're going to do a joint question session.
No worries.
And that means currently we're going to hear from our next speaker. So I'll just pull up an introduction for Juan. So Juan is originally from Colombia and just submitted his PhD in the School of Chemistry at UNSW. So congratulations, Juan. He's working under the supervision of Dr. Laura McCamish, and his research focuses on using computational molecular spectroscopy to calculate vibrational spectral data for thousands of potential biosignature molecules, some of which have incomplete or completely absent spectral data available. So with observations from the James Webb Space Telescope, already providing fascinating insights into the chemical inventory of exoplanetary atmospheres, Juan's data is playing a fundamental role in complementing the identification of new molecular species. Away from his research, Juan is also passionate about airplanes, outreach, and science communication, striving to facilitate scientific knowledge to the broader community. So whenever you're ready, Juan, you can pull up your slides and share with us.
Right. I'll just do this. Yeah, right. So is the slides okay? Are the slides okay? They're showing okay?
Yes, they're showing.
Okay, cool. Terrific. Thank you so much for that very kind introduction. As Rebecca said, my name is Juan. And yeah, so I just submitted my PhD, and today I'm really excited to share with you like the main findings and the whole story behind my PhD. So this presentation is pretty much on how we can use computational quantum chemistry to help find new molecules or to help find molecules in exoplanet atmospheres. So I always like to start with this little animation from NASA. So that was our solar system. And then all those dots that you can see there that look like a star, they're actually exoplanets, so planets outside our solar system. And I think a couple of weeks ago, we reached the 5,500 confirmed exoplanets. So at the moment, we know that there are at least more than 5,500 planets outside the solar system, which is completely wild. A very, very interesting milestone. And well, there's a lot of things that we can ask here. But the thing is that the closest exoplanet to Earth, I think is around 4 light years away. So with the current state of the art technology, there's no way we can go and visit those planets. So instead, if we want to answer question about exoplanets, we need to rely on what's happening in their atmosphere. And And in fact, there's a lot that we can learn from the atmosphere from looking at the atmosphere of exoplanets. So in this little cartoon, you can imagine this, I don't know, exoplanet, and then that planet hopefully will have an atmosphere, and in that atmosphere, there's going to be some molecules, right? So molecules will be making up the chemical inventory of that exoplanet atmosphere, and we can answer different questions about, or we can ask different questions about how those molecules make it into the atmosphere of that planet. So one thing could be volcano-related activity that is releasing these molecules into the atmosphere. There might be some geology events, so like geysers or things like that could also release these molecules. Weather-related events like lightning could also get molecules into the gas phase and make it into the atmosphere. And even potentially life, right? So I don't know, some type of non-intelligent life could be producing gaseous molecules as part of their metabolism, and those molecules could likely make it into the atmosphere. So there's a lot of insights that we can get from studying these exoplanet atmospheres. Now, we're sort of like venturing in this new phase of space exploration with the James Webb Space Telescope providing fascinating information about exoplanets. So we're actually looking at exoplanet atmospheres in a way that we had never done before. So one of the purposes of the James Webb Space Telescope is just, well, characterize the chemical composition of exoplanet atmospheres. And I think it's doing a lot more, but I'm just interested in this little bit. The frequency range covering from that the James Webb Space Telescope covers, it covers a really big part of the infrared, which is pretty cool because well, in the infrared is where when molecules interact with infrared radiation, they start vibrating. And those vibrations are unique for each molecule, so we get molecular fingerprints. So the fact that the James Webb Space Telescope is looking at this particular like region, will actually tell us a lot about the molecules that could be present in different atmospheres. And it has a much better resolution than previous telescopes, for example, Hubble and Spitzer. So yeah, fascinating things happening here with the James Webb Space Telescope in terms of analyzing the chemical composition of exoplanet atmospheres. But so we have the instrument, so the James Webb Space Telescope, to look for these molecules, but there's something that it's really fundamental here to actually make those detections, and that is the availability of high resolution spectroscopic data. So basically this means having all the different vibrational and rotational frequencies for a molecule as that molecule interacts with light. And so to gather these high resolution spectroscopic data, this is the result of the synergy between experimental and computational work. So on one hand, we have the computational chemistry part of generating this data, which means running very expensive calculations, very high levels of theory to produce potential energy and dipole moment surfaces that are really important to get sort of like the intensities of the different lines in the spectrum for a given molecule. And then the experimental spectroscopy part, which is literally just, taking your molecule to the lab and shooting some lasers to it to see how the molecule interacts with it. So from that experimental side, we get raw vibronic resolution with sub-wave number accuracy. So it's like really, really accurate. And so we need these sort of like two worlds to get the whole picture of what the spectroscopic data for a given molecule looks like. And then once we get that data, we can use it to compare with observations from the James Webb Space Telescope to actually decide whether a molecule is present or not in a given exoplanet atmosphere. One thing to notice here is that generating this data is really difficult and takes a lot of time, which means that approximately less than 100 molecules with high resolution spectroscopic data. So that's just like a very limited amount of molecules that we could say, yes, there's this complete spectrum of data for them. And just for reference, it took over 25 publications to obtain the spectroscopic data for TIO and a whole PhD, so approximately 4 years, to obtain the spectral data for phosphine. So these are just examples showcasing how difficult and how time-consuming it is to generate this data. And remember, we only have that data for approximately 100 molecules. And the big thing here is that if we don't have a spectroscopic data for a given molecule, then we simply cannot make any detections of that molecule. So we need that spectroscopic data for the molecule. So in other words, I think what this slide is saying is that we can, given our current state in generating spectroscopic data, we can only find about 100 molecules in exoplanet atmospheres, because those are the molecules for which we have the spectroscopic data for. But if you think about chemistry, well, chemistry can be way more complex than just 100 molecules. And when you look at the sort of like the demographics of these exoplanets, we're talking about ocean worlds, lava worlds, exoplanets where there's glass rain, exoplanets that orbit really close to the orbits and things like that. So these are very extreme environments. Sometimes these are very extreme environments where surely this really interesting and complex chemistry happening that is going to produce more than 100 molecules. And if we look at the astrobiology side of things, we can think how many molecules are involved in different like biological processes. And I think Satasiga and co-workers recently published a paper where they listed around 16,000 molecules that could be related to life, so as potential biosignature molecules. So there's a lot here. So chemistry can be more complex than just 100 molecules, but we just don't have data for the other molecules that could be also present in these planets. So the whole idea in my PhD was, okay, so how about we use computational quantum chemistry, so a standard quantum chemistry calculations, but in a high throughput fashion, to generate approximate vibrational spectral data rapidly for thousands of molecules. So this means not generating this really high accurate data as before, but instead generating approximate data somewhat like a little bit, not that accurate, but we can generate it for thousands of molecules. And this could be very useful to complement the search for molecules. So there are different advantages advantages from this approach. One of those is that if we calculate all this data for thousands of molecules, we can characterize a spectral congestion for different molecules. So if you think about a frequency window, how many molecules will have like a band or will absorb in that space? We can identify the strong molecular absorbers because as part of the calculations, we also get the intensity of the different spectroscopic lines. So you can just sort of like query this whole data set and ask for molecules that have like really strong intensities. And one of the most attractive things from this data is that we can find new molecular candidates to unknown spectral features. And this one here is very important. And that's something that I'm going to showcase later in this presentation, because I think it's one of the most powerful aspects of this approach. But the caveat here, and this is something that I really want to convey, is that the data that we've produced in here and that I've produced in my PhD is not accurate enough to allow any molecular detection in space. So it's not like you can get this data and compare it with a spectra from the James Webb Space Telescope to find molecules, because the accuracy doesn't work there. So this is a complementary approach where we can find potential molecular candidates, but we still need that high resolution data to make definitive molecular detections. So now when I talk about approximate vibrational spectral data, and now I'm gonna get here a little bit into the quantum chemistry side of this presentation. what I mean is harmonic vibrational frequency calculation. So just calculating the infrared spectra for different molecules. And just for the basics of computational quantum chemistry, there are a few things that you need to run any quantum chemistry calculation. The first one is what we call the method or the level of theory, which is basically the kind of approximations you use to solve the Schrodinger equation. So some of you might be familiar with words like B3-lib, Hartree-Fock, CCSDT, things like that. So those are just acronyms that enclose the different approximations used to solve the equations that arise during the calculations. And the other thing that you need in to run any quantum chemistry calculation is a basis set, which is basically how you represent electrons in computers. And so you might be familiar with things like 61G star or some of these acronyms again. That's just how you represent electrons. And the thing is that there are hundreds of basis sets and hundreds of levels of theory. So when you run any quantum chemistry calculation, you need to ask the question, what basis set and what level of theory or what method should I use, and how accurate my calculations are gonna be. And so we can sort of like get an approximate diagram of how the accuracy and the computational time like relate. So in this x-axis is the basis set size. That basically means, so the further to the right the basis set is, so say here, so the larger your basis set, that means your calculation is more accurate. You represent electrons better. And again, with the method class, so if the method sits up here, that means that the method is more accurate. So it will provide you better results. So ideally, you want to choose a method and a basis set that sits sort of like in the diagonal of this diagram, where you get a greater accuracy. So say here at the top, but also at a higher computational cost. So basically, you want to find the best combination that gives you pretty accurate results with like affordable computational timings. So I did that during my PhD, and it actually took a lot of time, sort of like benchmarking different levels of theory and basis sets to find which one was the most appropriate for running our calculations. And so this is just this table, and I'm happy to answer questions here if anyone has any. But just to summarize this here, we find these different methods and basis sets for running infrared calculations. And this one here particularly was pretty good because it represents a good compromise between accuracy and computational time for running calculations in a high throughput fashion, right? So like for running calculations for thousands of molecules. So the median error in the calculated frequencies is around 10 wave numbers, which again is not great. The accuracy is not that good, but it's not the worst thing that you could do. And the computational timings are pretty good. So all good. We have our computational methodology sorted. We know which method and basis set used to run our calculations. We know what our errors are going to be approximately. So the next question was, okay, so if we have this ready, how should we start running the calculations? So like which molecule should we consider? And so for a working set of molecules, we have our the chemistry periodic table, which is pretty good. But then we thought, which ones are the elements that are actually relevant for biologists? Like if we care about finding life on other planets, like life as we know it here on Earth, then we can get to the biologist periodic table, where you just have like maybe the most important biological elements: carbon, nitrogen, oxygen, phosphorus, and sulfur. And so we decided to work with sort of like this subset of molecules, and we ran these calculations for more than 2,700 SNOPS molecules. That's how we call them. So molecules containing only those elements. And all of these molecules are potential biosignatures from that work of Sarah Seeger and co-workers. So we had our methodology, so our quantum chemistry methodology, and the list of molecules that we wanted to get approximate spectral data for. And the thing here, the really important thing here is that from those 2,700 molecules, a really, really big part of these molecules, probably more than 90% of them, had no spectroscopic data available whatsoever. So you couldn't find any paper relating to how the different vibrational frequencies in these molecules are and things like that. So actually, by running these calculations, we're just producing the first available spectroscopic data for most of these molecules. So how did we go with this? So as I said, we had our methodology sorted. So we know what method and basis set to use and what our errors are approximately. And we have our list of molecules, right? So like the Schnapps molecules with around 2,700 molecules. So the next natural thing to do was, okay, let's run the calculations. We have the methodology, we have the molecules that just run all the calculations. We did all of that. And then we basically store all the computational data in sort of like a database and a data set. And I mean the data set, to be fancy, is actually a massive CSV file. But the thing here is that we basically have approximate vibrational spectral data, so infrared data for 2,743 Schnapps molecules, which again, most of them did not have any spectroscopic data available before. So pretty good. We have our data ready. So the next question was, okay, so how are we going to use it? And how are we going to showcase its applications? And so last year, the James Webb Space Telescope recorded the first atmospheric detection of CO2 on an exoplanet. So this exoplanet is WASP-39b here. And so the spectrum that you're seeing in the slide is the actual spectrum recorded from James Webb from this exoplanet. So you have all those lines. And basically this line here, that shoulder at like 2,250 wave numbers corresponds to the asymmetric CO stretch in CO2. So that one was the one that that confirmed the presence of CO2 on that planet. And that was pretty cool, exciting things. But when there was the press release of this discovery, my supervisor and I were looking at at their website, and then so they were only talking about how CO2 was present there, and then you can see that there's sort of like another shoulder here. And my supervisor and I thought, okay, so that's definitely another molecule. They're not talking, they haven't talked about it, so maybe they haven't found it yet. So let's see what our data can tell us about that particular line and whether we can come up with a list of candidates that could be responsible for that line. So that on non-molecular band at the time that they released this paper, or not the paper, but the press release. And so how we went about finding this molecule. So the first thing is that we chose a frequency window. So you can see that sort of shoulder is between 2,400 to 2,600 wave numbers. So literally that could be, that would be our frequency window. The next thing to know from the spectrum is that there's no apparent signal like here. So you can see it's sort of flat, or here either. And those two are really important like regions in infrared spectroscopy, because at 3,000 wave numbers, that's where the CH stretch vibration happens. So in like organic like molecules. So the fact that there's no signal at 3,000 wave numbers, that sort of work for us in the sense of telling us like, oh, the molecule might not have a CH stretch because there's no vibration there. And similarly up here, so the NH stretch is not present at 3,300 wave numbers. So again, maybe the molecule doesn't have an NH stretch. And the other thing is that WASP-39b is a really hot exoplanet. So we thought if there are molecules in the atmosphere of this exoplanet, there must be small molecules given the really high temperatures here. So these were all of like our constraints. We're looking for molecules there. And so we apply all of this to our CSV file to literally just ask, okay, so given these constraints, which molecules do you think will be responsible for this line here? And so this is what we found. So like the lines in yellow are our quantum chemistry data, and each line here represents a different molecule. So you can see that there are lots of molecules there. These are some of the examples of the molecules that we found were really good candidates here. So, things like SO2, ozone, SO3, so really interesting molecules. But, as it happens in science, a week later that we did this, they actually found that it was SO2, the molecule present there. And so this is from the paper that they published, where they made all the really robust analysis, and they found that it was SO2. But that's okay, because I think what this showcases is, so I'm not going to go back the previous slide, but basically, if you remember, these were the molecules that we found as potential candidates, and we actually found SO2 to be one of our potential molecular candidates there. they just confirmed it a week later. So this is pretty cool because it's literally showcasing that application that I talked about before, where even though this is approximate data, we can still provide a list of potential molecular candidates that are worth exploring and worth studying more when we find these unknown spectral peaks in a spectra. So pretty cool. And then we got kind of like another chance. So this is from that same spectrum. So, you know, there's the CO2 detection, the SO2 detection. They also found water. But then there's this feature here at 4.56 micron. And this is like an extract from the nature paper. You don't have to read it, but basically what they're saying there is we found this peak and we tried every single molecule for which we have high resolution spectroscopic data for, and none of them could reproduce that line. So basically, in other words, what that is saying is because we don't have the spectral data for this molecule here, then we simply cannot find that molecule. Which again, you know, it goes back to pretty much the motivation behind my whole PhD, and is, okay, so we're just limited to finding these 100 molecules because we only have data for them and not for the other ones. So cool. So we asked again, okay, let's go about finding molecules, finding a list of molecules responsible for this unknown line. So we did the same, you know, frequency window in this case, 2100 to 2300 wave numbers, no CH, NH trash, hot exoplanet, small molecules. And so, yeah, so this is sort of like where the unknown band is. And we found, again, a couple of molecules there. Each line is a different molecule, but there were 3 molecules very interesting. So H has SCN, C2N2O, and CHNO, were the 3 molecules that like caught our attention. This is just a table showing the data for these 3 molecules. So, sorry, I'm just gonna go back. Remember that the line that we are looking for is at 2100, like 2192 wave numbers. And for these molecules, yeah, so they're pretty close to that sort of like unknown line. And all of these molecules have been found in the interstellar medium. So these are 3 molecules that are really interesting at the moment. And we're sort of like working with astronomers to find ways of testing if these molecules are actually present there, which could be really interesting. Again, just a list of candidates. But there are like more fundamental questions that we need to ask with this, because matching the spectroscopic data with the data from the James Webb Space Telescope is just the first step. We need to ask more questions about what are the reaction and destruction pathways in that given exoplanet atmosphere, whether there's high resolution data, and what is the likelihood that these molecules will actually be present in this exoplanet. So there's a lot of research that we need to do here, but we're sort of making progress towards the first step. Just a little bit more here on sort of where we're going with these molecules is, so again, this little part here, it was the one that we got from the James Webb Space Telescope. So there are different lines for these molecules, but we know that these molecules have more vibrational lines in different regions. So like down here. And so if we use the MIRI instrument in James Webb, then we could look for these signals, and that could be more, that could basically lead to a more robust identification if these molecules happen to be the ones responsible for that unknown line. So this is a still work in progress, and we'll see how we go there. Sort of like the future for this work, there's a lot of things that we need to do here to like improve the quality of data that we're having here. But one of the most important things, one of the most important things to address for the future is how are we going to include approximate raw vibrational profile to the frequencies. So at the moment, one of the big limitations in our data is that we literally just have line spectra. So that means we have the number for the vibrational frequency and the intensity. And that's why we only have like these lines here. But in reality, the infrared spectrum for a molecule doesn't look like single lines, but it has a shape to it. And that's because you cannot separate vibrations from rotations. And so the rotations in the molecule are the ones that give the different spectral lines that sort of like shape to it. And so it is really important when finding molecules because it definitely changes the way the bands look like, and they're really dependent on temperature. So this plot here just showcases that. So again, the data in black is the one from the James Webb Space Telescope. So this is the line for SO2, and you can see that how different the same band looks for between our data, which is the one in yellow, and the high resolution data available, because the high resolution data has the shape of the bands. So it looks very different. And then between the two high resolution ones, you can see how the temperature has a really big influence. So if you have it at 100 Kelvin, it looks like this, but at this temperature, which is the temperature of the exoplanet, it actually takes the actual form of the band that was found on the exoplanet. So we're sort of like working on how to incorporate these approximate raw vibrational profiles here to actually test them in the different models that account for these molecules in exoplanet atmospheres. Sort of the dream here. So basically our approach is we want to generate these approximate spectral data for thousands of molecules, and that's sort of here where we are. And then from there, we can provide directions into which molecules require high resolution data. It's better to start from somewhere, right? So I don't know if I tell you like, oh, we need high resolution data for these 5 molecules. It's better than just going, we need high resolution data. Where should we start? So we're sort of providing directions there. And then once we have that high resolution data, we can do lab spectroscopy for those molecules, and ideally get astro observations of molecules. So basically just finding molecules in space. And again, my PhD on the work that my supervisor, Laura, and I did pretty much sits at the beginning of this whole pipeline, and is, okay, let's produce high throughput approximate spectral data for thousands of molecules and see what we can do with them. But just in the more like general space, so looking for astrobiology beyond our solar system, so in exoplanet atmospheres, means looking for molecules in planetary atmospheres. And this is a community effort, right? So we have, the exoplanetary atmosphere observations with, say, the James Webb Space Telescope, and that we use those to get some atmospheric models and find the molecules present there. And maybe that could mean aliens, like once you've sort of like addressed the whole pipeline. But there's a lot that comes into play. So in these atmospheric models, we need the physical and chemical characteristics of the atmosphere to sort of like make them robust and actually account for the different effects that will produce or remove molecules in the atmosphere. We need molecular spectroscopy, fundamental in this process. If we don't have spectral data, we cannot find a molecule. But we need the astrobiology and origins of life research to sort of like determine whether a given molecule actually represents life, or is just, like a false positive in the sense of it could be produced by geology or something like that. So it's like a really, big community effort with input from different disciplines, which is pretty exciting. And yeah, I just would like to finalize just thanking my chemicals group. We're a quantum chemistry and a molecular spectroscopy group at the University of New South Wales in Sydney. So that's the current state of the group. That's Laura, that's me. And I also thank NCI, which is the National Computational Interface, for, giving us the computational timings for running all these calculations. And yeah, that's pretty much it. Sorry, I think I went over time, but thank you.
You're absolutely okay. We still have about a half hour for questions and follow-up and discussion afterward. And that was just a really beautiful talk, also with really beautiful slides. Thank you for that.
Oh, thank you. Thanks.
So right now is a question session for both you and Lauren. So I'll invite her back as well. And while you were talking, Juan, I did see some questions roll into the chat for you, so we could start there. However, they are in Spanish, so I may not be able to translate them to ask the questions, and I may rely on you if you are able to translate the questions that came in and share your answers to that.
Yeah, sure. Let me just, my Zoom is acting up, so okay, cool. I'll just have a look at them.
And while you're looking, I'll just say it's really awesome to have a broad audience like that. And I think maybe you brought that audience with you. So thank you. And we're excited you're here, everybody. I can try to read them, but
No, that's all good. I think one of the questions is about whether there could be methane in these like sort of list of potential molecules, and the answer is yes. Methane is a potential biosignature. So it is one of the molecules that we have calculated and that we have in our database. So basically, we could use it to find other peaks in to test unknown peaks in exoplanet atmospheres. The thing that I want to say there is that methane is one of it belongs to that list of 100 molecules that we have high resolution spectroscopic data for. So there's actually better data for methane than the one that we produce. So actually, if people find methane or lines that belong to methane in a given exoplanet atmosphere spectrum, they can definitely use the really high resolution spectroscopic data to match those observations. So yeah, but definitely methane is there.
there's, I think, one after that.
Yeah. Sorry, I'm just.
In what ways could you discover some other molecules? Something along those lines.
Yeah. So it's like yeah, it's like some I think it's just the same way. So like when the James Webb Space or different space telescopes actually gather this data from exoplanet atmospheres or planetary atmospheres in general, we get all the peaks from like all the peaks from the molecules that are present there. It's just a matter of having the right spectroscopic data to match the observations. So I think that's pretty much like that's how I understand the question. Yeah, but I'm happy to go, I don't know, to provide a better explanation if that helps.
Yeah. Did you put your email up? I can't remember.
No, I did not.
You don't have to. You don't have to share.
No, that's all good. I'll just put it in the chat in case. Yeah.
And there's another question in the chat for you again, Juan. And just remember, we can put all questions in the chat, or I can also allow people to speak if they want to raise their hands. We can unmute you. So that's something you could do as well. Okay, so the next question I see is that the astronomy community recently began to be interested in technosignatures. Do you have any thoughts about technosignatures?
Yeah, 100%. Just in case, I don't know, so technosignatures, in case someone is wondering what they are, so it's basically signals of intelligent life. That's how I would like to describe it, like on exoplanet or on different in space in general. So you can think about, for example, like Wi-Fi signals or light pollution from like cities in other planets, on other planets. So that's what they mean about technosignatures. And yes, in the case of in the sense of molecules, we can think when we think about technosignatures, we can think about, for example, CFCs. So those are molecules that are not naturally produced, but they were human made. So if there's another civilization that is also, destroying their atmosphere, producing CFCs, I know with the current telescopes like the James Webb Space Telescopes and new observatories that will be launching soon, we have the capacity to find those molecules as well. So that is like sort of like a pathway that we're sort of taking, is like actually looking for CFCs or other pollutants that could be present in the atmosphere of different planets, and tell us about civilizations there. In terms of my work, we only have the spectroscopic data for these SNOPs molecules, and so CFCs are not there because we didn't consider molecules with halogens. But that's definitely an expansion that is currently happening in our group. So we have different students that are looking at how we can expand this sort of data set to include a more diverse range of molecules and actually look for technosignatures.
It kind of makes you wonder if anyone out there on an exoplanet is discovering us by our pollution.
Yeah, 100%. Yes.
Yeah. So I'll ask a question to Lauren, which is that you're discovering how finally the pH influences the properties of these different protocells and vesicles. And I'm wondering about the early Earth environment and how stable pH would have been at that time. So I maybe have a general question of what do you think swings in pH maybe on early Earth in a not so stable environment, how going between different pHs might influence the properties of the vesicles?
Yeah, great question. I would say generally, like we would probably expect there to be much greater swings than what just a plain fatty acid vesicle can actually withstand. So generally on their own, they're only usually stable around 1 pH unit, kind of around their pKa, maybe a little bit more. But you can incorporate like other molecules into the membrane. There's different ways to actually, I guess, stabilize them to a wider range of pHs. But generally, even within that pH range, if we're say like swinging from one end to the other kind of thing, I would suspect that you can kind of get somewhat of like even like an oscillating system, for example. And that's kind of something I'm working on a little bit at the moment. Seeing like, we can maybe trigger a chemical reaction that leads to an increase in pH, which might lead to like some of those reactants entering or exiting the cell. And then what happens if the pH goes down again? Maybe you block off that reaction from occurring or something like that. But yeah, I suspect like within the pH range at which vesicles kind of form, you can kind of just see those properties sort of oscillate a bit and just swing either way.
Yeah, it might be interesting, and we'll be interested to see that future work about your
calculations. Yeah, it's a work in progress.
Let's see, I'm gonna catch up in the chat. So we have a question from Kaden to Juan. So it's, how do radio telescopes play into this, or do you only use infrared?
Yeah, that's a good question. From my understanding, I know like JWST and other telescopes are pretty much looking at the infrared when it comes to exoplanetary atmospheres. I know radio telescopes play a very important role in finding molecules in the interstellar medium. So, the medium between stars and stuff. So yeah, I think they're just like working on different domains in space. But still like because it's really important, right? Like that they both work together, because if you find a molecule in the interstellar medium, you need to take that into account when looking for molecules in planetary atmospheres, because the interstellar medium is gonna still be there between this telescope and the planet. And so you need to make sure that the sort of like the signals don't interfere between them, and then you sort of like claiming to see a molecule in an exoplanet atmosphere, but you actually seeing it in the interstellar medium and not in the planetary atmosphere. Yeah.
Thank you for that. The next question I see is for Lauren from David, which is, are these ionic permeabilities selective? In the same line, have you tried to measure anionic permeabilities? And have you tried to add membrane proteins to the vesicles? So 3 questions.
I guess the first one, I haven't explored that myself personally. There is other work that already exists that kind of shows that different ions can affect the properties of vesicles in different ways, of fatty acid vesicles in different ways. But again, it's not something I've particularly explored with this system in regards to permeability, and same with the anions as well. I haven't personally explored that. It's a bit trickier to look at. In regards to the membrane proteins, I haven't tried adding them to these fatty acid vesicles. I've been working a little bit with phospholipid-based protocells and membrane proteins, but I haven't really tried incorporating membrane proteins into fatty acid-based systems.
I think that was all the questions. Yeah,
I think so. Yeah, but the membrane proteins it's a tricky one. You would expect that would have a significant effect on membrane properties, and it definitely does for phospholipid vesicles.
Maybe I'll pop in another question similar, which is how membranes in full-on cells might respond to very small changes in pH based on what you know, or maybe the literature.
Yeah. So generally, so for the most part, like our cells are primarily composed of a phospholipid-based membrane. Generally, they are nowhere near as susceptible to these pH changes as the fatty acid ones. So they're a lot more robust. They can tolerate a much wider range of pHs, for sure, which is probably ideal in terms of an evolution point of view. Yeah, you definitely don't see this, I guess, these extremes in terms of these property changes just over the small range. You do see some similar trends in terms of, say, like the conductance readings that I looked at, for example. You do see a similar trends with increasing pH for phospholipid-based systems, but it's over like a much wider pH range. That's like, I don't remember off the top of my head, but like pH 3 to pH 10, you maybe see a little bit of an increase rather than just looking at like 0.5 or something like that.
I guess they're highly evolved or something.
Makes sense.
Yeah. Let's see. Sort of lost track here. I think 2 questions in the chat again for Lauren. So do you think that being able to analyze pH levels on, well, there maybe is a relationship here. pH levels on exoplanets could help identify environments stable enough for early protocells to live? Interesting.
Yeah, great question. I definitely think so. I think it's like as much information we can obtain about like some of these exoplanets, like I think that's definitely for the best in terms of like promoting, I guess, directions for like where our research should be going in terms of like potential origins of life scenarios. Obviously, like some of these fatty acid based systems just on their own, like if you've got a crazy low pH, for example, like it's probably not really going to work out. We need to explore other, I guess, potential options in regards to what protocells would actually be stable in those kind of environments. So I definitely think it would help, I guess, direct people in this community as to like what direction their research should be taking, for sure.
What about you, Juan? Do you have any input on the pHs of these different environments?
Yeah, I think it's a really interesting question. I guess the first thing that came to mind when you were reading the question was, I don't really know how would you go about like measuring the pH levels on an exoplanet. Like I think that's probably the most challenging bit. Because I honestly don't know. Because yeah, all the information that we can gather so far comes from what we see in the atmosphere. And like one of the limitations that we're facing right now with current telescopes is that we can only see so deep into the atmosphere actually. So when we look at these molecules and stuff, they're pretty much in the upper layers of the atmosphere. So like we're not really like penetrating that much. And definitely I'm assuming pH levels would change drastically from the upper layers in the atmosphere to what's actually happening on the ground if you have a rocky exoplanet. But if you have a gas giant exoplanet like WASP 39b, for example, then yeah, I feel like the further in you go into the atmosphere, the changes will the pH will also change. So that was the first thing that came to mind, was how on earth would you go about measuring pH levels? But yeah, it's a really interesting question. Yeah.
Jumping back to Lauren, the next thing we have in the chat is what about osmotic environment properties, salts, etc., and varying fatty acid chain lengths? How does that impact your vesicles?
Yeah, both of those things definitely impact vesicles. One of the big things about varying fatty acid chain lengths is it actually affects basically the minimum concentration you need for fatty acid vesicles to form. So as you decrease the chain length, rather, you actually need a much higher concentration of that fatty acid to get vesicles to form, which is kind of unfortunate, I guess, because you would expect that much smaller fatty acids would be more prominent on an early Earth environment. But the fact that you need a lot more of them to actually make these vesicles is a little bit of a shame. But in regards to osmotic environment as well, yeah, that definitely affects things. So things like whatever salts around, whatever cations, especially like the presence of divalent cations definitely affects these vesicles. And even things like changing osmotic pressures can also definitely affect the behaviors of these vesicles. And you can see some pretty interesting behaviors in regard to just the changing environment again in general, I guess.
I might, sorry, I'm peppering in questions since we spent time in between other people's questions. But I noticed in your slide about what you're doing in the future that you pointed out you're doing studies with some divalent metals and iron, I guess trivalent iron. And I'm just wondering maybe what's the most surprising or interesting thing you might have found so far?
Yeah, I think so far out of it's probably still early days, but so far out of what I've looked at, I was very surprised to see that copper didn't really seem to do a whole lot to the vesicles. It's generally, it's kind of somewhat assumed that most divalent cations have a pretty negative effect on fatty acid vesicles. And you see that like from day to day with things like soap scum, just like different divalent cations interacting with fatty acids. It's generally not ideal. And you definitely see that with something like calcium and magnesium. It just obliterates the vesicles depending on what concentrations you're working with. But I was able to go to pretty relatively high concentrations of copper, and it didn't seem to be doing a whole lot in terms of just like overall vesicle kind of morphology, just how they actually looked. So I think that was kind of surprised. That was pretty surprising to me, especially given like the other divalent cations.
I'll fill in with the background for just people maybe wondering why ask that question or why do that research, just because life has such a dependence on metals. So we want our containers to be compatible with the metals.
Yeah, indeed.
Yeah. There's a question from Caden, again for Juan, and he's also noting, Caden is noting that he's a potential future astrobiologist, so welcome to the community. And he's asking, or saying that you mentioned the importance of evolutionary biology to astrobiology. How would that be incorporated?
Yeah, that's interesting. I don't remember mentioning evolutionary biology, to be honest. But I think, well, that's a difficult question. I think probably just studying, like, because, sorry, what I'm thinking here is like in terms of exoplanet atmospheres, because we can, like, so far what we're doing is analyzing the presence of gaseous molecules in the atmosphere of these planets, right? I think an interesting bit that could be related with how evolutionary biology could play in here is just probably like studying how the metabolism of different, I don't know, like a species evolves, right? Or has evolved. And then like which gases are being produced in each state, whether that's something that changes over time, or like with the different like evolutionary stages for a given species. And then so that could give you some insights into, okay, so if X gas is being produced in like really big quantities, and we can relate it to these species, then that can sort of tell us like where in its evolutionary stage is. But yeah, that's me guessing right now there. I honestly, I don't really see how it could be incorporated like that explicitly, to be honest.
But maybe generally one could imagine an evolution of an atmosphere with the evolution of the biology on the planet.
Yeah, that's true. Because definitely you can get a lot of insights from the evolution of the planet, like the planetary evolution, sorry, the planetary like formation, I guess, by looking at the molecules in the atmosphere. So you can get definitely a lot of insights from there. Would be just how you relate it to the biology, if there's biology happening in that planet, on that planet.
And next we have a question for Lauren. And AC says, from the evaporation pools prebiotic origin theory, would that help in concentrating the shorter possibility of more abundant fatty acid chain species?
Yes. Short answer is definitely yes. There's been a few people who've done experiments pretty much kind of showing those processes and how they can be, hot spring theories can be advantageous for fatty acid-based protocells. There's a colleague in my lab who's got a paper published that's kind of related to that concept. Definitely yes.
Nice. Short and sweet. I like clear answers.
Yeah.
All right. So I have a question for Juan, which is, and I'm drawing a parallel to in biochemistry, how we can create models of the ways that proteins should fold, and it helps to actually resolve the structures from the model and make that resolution process a lot faster and easier. So I'm wondering if in your case, when you make predictions of spectra, if it does the same thing or something similar where it actually speeds up the process, the science of resolving the spectra from the—
You mean like when comparing it to like telescope, like from the telescope data? Is that what you mean?
What I mean is, you mentioned at the beginning of your talk that it's a long process and a hard process to actually find the, I suppose, true spectra of different molecules, but here you are predicting them. So I'm wondering if your prediction does anything to make the actual solving and the finding. See, this is where I'm reaching my limit, but the actual getting to the spectra faster or easier.
Yeah, no, that's a really good question. I think that there are like different things that come into play here. The one that is probably the most important is that when I was talking about this really high resolution bit and how it takes a lot of time, one of the reasons why the computational part, so the experimental part is just difficult, so there's that. But one of the reasons why the computational part is hard or takes so much time is because it is actually calculating every single transition that the molecule can undergo. And so that includes not only the vibrations, but all the rotations that the molecule has. So it basically means that you need to run a lot of calculations and like guess a lot of like the transitions between the different states. In the calculations that I perform, they're just an approximation to those really high accurate calculations. So the first thing that we do not do, that they do, is calculating these rotational bits. So that's why I was saying at some point in the presentation how we only have like lines, like vertical lines instead of bands or shapes. And that's because we don't calculate anything that will give that band the shape. Whereas the really high accurate calculations they do calculate every single peak, and then they give you like that sort of shape. So there's definitely that. And the other thing is that the calculations that we do, they're just they not only they calculate like that vibrational frequency, but they're also an approximation. And so we calculate only a given number of frequencies, which are not the most important, but most of the time the most prominent vibrational ones. But there are other frequencies that need to be calculated that they're definitely beyond the scope of our calculations. So we only get so far into those calculations, but what we get is like good enough to get us started. I don't know if that sort of answers your question.
I think so. So when we have a complete spectra, I suppose, that's also computationally calculated versus measured physically? Or—
Yes. Yeah. So they complement each other actually, because with the experiment, you can get like you get okay. So there are two things that sorry. The first one with experiment is that you get regions of the spectrum, but there are other regions in the whole electromagnetic like spectrum, say, or range, where doing the experimental part becomes more challenging. And so there are like, if you look only at the experimental data, there are like gaps, like really big gaps there. And the other thing with experiment is that you can get really, really accurate frequencies. So, sub-wave number accuracy. So literally I don't know, frequencies that deviate in like 0.01 wave numbers from where the actual frequency is. So really, really accurate. But the intensity of those lines is extremely difficult to calculate experimentally. And the accuracy is really bad. So you get really good. So you sort of like experiment can give you really good answers in the x-axis of the spectrum, but in the y-axis, how intense those lines are, it's really, really like, yeah, difficult, sort of say. And then that's where the computational part comes in, because computationally we can get really good intensities. So the intensities come there, and so we complement that. And we also, with the computational part, complement the gaps that you couldn't calculate experimental. Sorry, not calculate, but perform experimentally. So yeah, it is a synergy. We need both to get the high resolution data.
Great. Thank you. Hopefully more people than just me benefited from that, but that was helpful.
Yeah.
There's another question in the chat. I'll leave it with that one since it's about almost 6:30. So the last question we have here is that the isotopic element concentrations could vary in some exoplanets. So this could alter the spectral properties of some of the CHNOPS gases, and I'll just fill in CHNOPS for the ones you mentioned that are important to life: the carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur.
Yes, 100%. So isotopic changes, they will definitely change how the spectra for a given molecule looks like. Especially like, yeah, if you think about carbon-bearing species, so if you have like carbon-13 instead of carbon-12, like that will definitely shift some of the lines. The thing here, we're sort of addressing that and not really at the moment. And one of the reasons why is that even though the James Webb Space Telescope is providing, amazing resolution compared with Hubble and Spitzer, where that is still not like the resolution is not, it's not really high enough yet to differentiate between isotopic species, especially when considering those shifts in the frequency. So at the moment, I think, yeah, it was sort of I don't know, like in a gray area maybe, where like, yes, they do have an effect, but I don't know if we are actually able to see it yet.
It'll be interesting to see that technology grow and that capability grow.
Oh, 100%. Yes.
All right. Well, I want to thank both of you again for your talks, really wonderful talks today. And I want to thank everybody for coming and hope you enjoyed it. And we probably won't see you in 3 weeks because of Thanksgiving in the United States. But we will see you sometime after that. Just stay tuned. Thank you, everyone.
Thank you.